excited to introduce our Craig, who we have as our speaker today. So he is an adjunct professor at the University of Massachusetts Chan Medical School, RNA Therapeutics Institute. Today he'll be talking about nucleic acid immunity and implications for optimizing cancer and immunotherapy. You may have met Art at some of the OTS meetings. He's a great guy to talk to. He did a ton of kind of live communication videos during the pandemic, so you may have also seen him there. And so I'll just remind you to please answer or ask your questions rather in the Q&A function, and at the end of the chat, we'll have a curated question session. All right, Art, you can take it away.
Okay, thanks very much. Appreciate that. And I'm glad to have the chance to talk with you all today and look forward to questions afterwards. I wanted to start by just laying out the goals of what I want to accomplish in this webinar on nucleic acid immunity and cancer immunotherapy. First of all, just introducing this concept of nucleic acid immunity, because some people aren't really that clear in it, and how it functions as an arm of innate immunity and host defense. And the key part of this that I'm going to focus on are the nucleic acid immune sensing receptors, their agonists, and their mechanisms of action in cancer immunotherapy. And that's going to lead into the differences between type 1 interferons and a type of immune cell that many of you have heard about, but a lot of people don't understand very much about, and that's the plasmacytoid dendritic cell. And then ultimately, I really want to inspire people to study these PDCs in whatever you're working on. If you're working on nucleic acid therapeutics for cancer immunotherapy, then it's in your interest to understand how those affect PDCs, and I'm going to explain why. And then some of you who are earlier in your careers and are thinking, what do I want to do? I hope I'll inspire. Yeah.
Sorry to disturb. It might be nice to put it on the presentation mode. Presenter mode.
Oh, I thought it was in presentation mode.
See the rest of the screen as well.
Okay, let's see. That's odd. How's that?
Yeah, perfect.
I just had to click on that. Okay.
3.
Thanks. No, that's good. And I hope I inspire some of you to study TLR7 and TLR9 agonists. And my disclosures here, feel free to email me with any questions. So nucleic acid immunity evolved at the origins of life, actually, more than 1 billion years ago, we think, in bacteria, to defend against bacteriophage and viruses that try to infect the bacteria, hack its genome, and introduce their own genes and force the bacteria to make more copies of the bacteriophage. And so some of the examples of these nucleic acid immune defenses are nucleases, DNA restriction enzymes, the CRISPR pathway. These are all forms of innate immunity. And as a reminder, innate means pre-existing. Immunity are defenses against infection. And so these innate immune defenses are characterized by acting rapidly to control infection. And there's a marvelous review article that Gunter Hartmann wrote some years ago on how nucleic acid immunity evolved to protect genome integrity. And Gunter made the point that organisms throughout biology need to maintain the integrity of their genomes. And nucleic acid immunity is how they do that. So in bacteria, we have these systems, nucleases, the restriction modification systems, CRISPR-Cas9. And then throughout evolution, some of these defenses were not maintained. Others were added on, like RNA interference and nucleic acid receptors. Now, restricting nucleic acid function. Now, these receptors in the green box can destroy or modify nucleic acids, but pathogens can evolve to avoid those by simply changing their nucleic acids. So these are somewhat limited in that sense. Nevertheless, they've been incredibly important to our field and to the whole biotech industry. Nucleases are what make antisense oligos work, or specifically RNase H. DNA restriction enzymes, we wouldn't have a biotech industry or molecular biology without those restriction enzymes. I don't need to remind you about how CRISPR-Cas9 design is used now, or how RNA interference is used to make siRNAs. And then RNA editing is the latest very exciting approach to use these mechanisms that evolved as for nucleic acid immunity for therapeutics. And of course, many of these mechanisms have evolved other functions since then. RNA interference no longer plays a major role in defending us against pathogens. Now our cells use it for other things, processing RNAs and so forth. What I want to focus on today, though, are the most highly evolved mechanisms of nucleic acid immunity. And those are the nucleic acid immune sensing receptors, which are characterized by their ability to induce the production of type 1 interferons. Now, these receptors have a key role in innate immunity to detect foreign nucleic acids and to initiate adaptive immunity. So this turns out to be a much more powerful way to protect our genome integrity using cells such as CD8 positive T cells, also called cytolytic or cytopathic T lymphocytes. These can kill viral or retroviral infected cells. And this is really an amazing level of specificity because a retrovirus may just have a couple of genes. And our immune system has never seen those genes before, and yet these cells can be induced to kill viral infected cells without damaging uninfected cells in the host. And of course, this is what we're interested in harnessing for cancer immunotherapy, because those same cells that originally evolved to defend our genomes can also kill tumors. Just as an aside, I want to point out that as powerful as these mechanisms are, they actually haven't done a fantastic job of protecting our genome integrity, because as you're probably aware, somewhere around half of the human genome, of all vertebrate genomes, has been derived from reverse transcription. And the percentage of our genome that is taken over, has been taken over by endogenous retroviruses is more than what's devoted to genes. Nevertheless, these mechanisms are still very, very powerful. And the whole idea of activating innate immunity for cancer immunotherapy really involves stimulating these receptors. So I'm going to spend most of my time talking about that. But first, just to quickly review the key concepts of vertebrate innate immunity. The skin is a very important part of our innate immune system because it keeps pathogens on the outside. And when that barrier is breached through whatever mechanism, the job of the innate immune system is first of all to detect that infection. And it does this using receptors for pathogen-expressed molecules. Now, in addition to detecting the infection, the innate immune system has to categorize the infection. It has to determine, is this an extracellular infection from bacteria or parasites, or is it an intracellular infection? And the reason that this is important is because once viruses have invaded our cells and they're replicating inside those cells, you need different defenses to eliminate those viral infected cells and clear the infection. And so the third part of innate immunity, once it's categorized the infection, and all of this occurs within minutes to hours, is to activate the defenses that are going to contain that infection and then clear the infection. And in the case of extracellular pathogens, the correct response to make to control that is inflammatory cytokines, TNF, IL-1, IL-6, and the cell types that play particularly important roles there are neutrophils. With intracellular pathogens, interferon alpha is arguably the single most important factor to restrict viral replication in the infected cells. And with both extracellular and intracellular pathogens, that early phase of the innate immune response that occurs within hours leads to an adaptive immune response over the following weeks to really clear the infection. And with viruses, the key parts of that are these CD8 positive T cells or CTLs that kill infected cells. And of course, you also need B cells to produce antibodies that will bind the virus and block it from infecting new cells. So what are some of the receptors that our innate immune system uses to detect and categorize these infections and then initiate defenses? The toll-like receptors are really the best understood family. There are other families, of course, so this is just an example of how these can work. And some of the toll-like receptors are on the cell surface where they appear to function to detect extracellular pathogens. So for example, TLR4 detects lipopolysaccharides. That's a signal to the immune system that there's a gram-negative bacterial infection. And the response to that, these TLR4 is expressed in monocytes and conventional dendritic cells, and that leads to an appropriate immune response with inflammatory cytokines. You don't need interferon alpha or CD8 T cells with an extracellular pathogen like that. Now, other TLRs are inside the cell, especially in the endosomal compartment. In the case of the TLRs, that's a great location for them to be because viruses enter cells typically through the endosomal compartment. And these TLRs are expressed in largely different but somewhat overlapping subsets of dendritic cells and macrophages. And I'm going to spend most of my time talking about TLR7 and TLR9 because these are the two nucleic acid immune sensing receptors that are expressed in a cell type called the plasmacytoid dendritic cell, or PDC. And this is the key cell for inducing the massive amounts of interferon alpha that need to be produced to fight off viral infections and induce the CTL responses. So if these defenses work, then why wouldn't the pathogens evolve ways to avoid triggering them? And in fact, they probably have to some degree. It turns out that the HIV genome is highly CpG suppressed. It has much less of the CpGs that would be expected based on random base usage. I forgot to point out here that TLR7 especially detects uridine or guanine uridine-rich single-stranded RNA, and TLR9 is activated by unmethylated CpG DNA. In our genomes and all other vertebrates, when a C is followed by a G, the C is normally methylated, and CpGs are suppressed in our genomes. They're also suppressed in the genomes of all retroviruses, small DNA viruses. In the case of HIV, HIV has 14% of the expected CpGs that ought to be present. This is just looking across the HIV genome. The red line shows the expected frequency of CpGs. And, why is this? There's no known mechanism to explain how CpGs could have been depleted from the HIV genome. And, maybe it's going a bit far here to infer from this that the CpG DNA is actually an effective antiviral defense. But if that is true, then activating that defense ought to be able to eradicate tumors if we do it in the same way that the retroviruses are. And conversely, if you engineered a retrovirus to have the expected CpG frequency, you would expect it to be much more efficient at stimulating these responses. And so such a vector might be useful for cancer immunotherapy. Now, this is a summary of the nucleic acid immune sensing receptors that have been targeted for clinical drug development in humans. Two of them are for DNA. I mentioned TLR9 already, expressed in the endosome. The agonist for this is unmethylated CpG DNA that's present in viruses and also present in bacteria. And in our genomes, these are methylated. cGAS sting pathway can be activated by self-DNA as well as foreign DNA. The way that we avoid this pathway normally being activated is that, of course, our DNA is normally in the nucleus, and this pathway is in the cytoplasm. Now, when cells are stressed, DNA can escape from the nucleus into the cytoplasm. So this probably has a role in homeostasis, but it doesn't distinguish pathogens from self. TLR7 and TLR8, on the other hand, both recognize uridine containing RNAs. And these do provide some differentiation between self and non-self, because the most abundant RNAs in our cells, ribosomal RNA, tRNA, the small nuclear RNAs, those use pseudouridines. And pseudouridine doesn't stimulate these receptors. So the viral nucleic acids will preferentially activate those. And likewise, in the case of RIG-I, and the RIG-I-like receptors in the cytoplasm, this is activated by structures, 5' triphosphate, double-stranded RNAs. Diphosphate can activate too. And these are also present in vitro transcribed RNAs. They're a common contaminant in mRNA preparations, mRNA vaccines. In the case of our own nucleic acids, they usually use 5' monophosphate, so that avoids triggering of this. Now, all of these pathways function differently in rodents and primates. Many of them are expressed in different cell types, or they activate different downstream pathways. And so it is not possible to rank order the efficacy of these pathways in mouse models and predict what's going to be most effective in humans. You really have to do the studies in human cells. So it turns out as well then that these different nucleic acid receptors are expressed in different cell types. Now some of them are ubiquitously expressed, like the cGAS STING pathway, RIG-I. Those are expressed in tumors as well, and they can have a variety of effects that can be therapeutically beneficial. Monocytes and macrophages express some of them in some cells, including TLR7. Conventional dendritic cells also. The plasmacytoid dendritic cell that I mentioned before is unique among these cells because although all of these cells can make type 1 interferons, the other cells predominantly make interferon beta. And the primary source of interferon alpha is the plasmacytoid dendritic cell. And this cell also has a key role in regulating tolerance versus Th1 immunity. The so-called Th1 response is the type of CD8 positive T cell response. And the only receptors that can activate PDCs to make that interferon alpha are TLR7 and TLR9. So why am I distinguishing interferon alpha from interferon beta? Given that these are both type 1 interferons, they both bind the same receptor, interferon alpha receptor. They bind it with different affinities, and they have different on and off rates. And it's very clear from human clinical trials that these have distinct biology. Interferon alpha is approved in humans for treating hepatitis C virus. It has efficacy in some types of cancer, but if it's given to patients with multiple sclerosis, classic T-cell mediated autoimmune disease, it aggravates the disease. Gain of function mutations in interferon alpha, or the pathways leading to interferon alpha, have been associated with classical systemic autoimmune diseases like lupus and psoriasis, and this CD8 positive T-cell response that I've been talking about. On the other hand, interferon beta has relatively little activity in hepatitis C virus, negligible activity in cancers, and it's approved for the treatment of multiple sclerosis. It ameliorates disease. So, to somewhat oversimplify, interferon alpha drives CD8 positive T-cell mediated immunity. Interferon beta ameliorates it. This is probably the single most important point that many people working in cancer immunotherapy are not aware of. And when I tell people this, they're surprised. And the implications are, of course, obvious, I think. For cancer immunotherapy, we need to optimize the induction of interferon alpha. Interferon beta, because it has a higher affinity, it actually might be interfering with the activity of interferon alpha. And the only pathways to induce maximal interferon alpha production are TLR7 and TLR9. And that's why I'm really going to try to convince people to look at those pathways and focus on them. So what are the immune effects of stimulating TLR9? TLR9 is really only expressed in two immune cell types in humans, B cells and plasmacytoid dendritic cells. And so at a high level, CpG DNA activates those cells. B cells secrete some inflammatory cytokines. They express co-stimulatory receptors that help them differentiate into antibody secreting cells. And if they're stimulated through TLR9, they have an enhanced response through the B cell antigen receptor if they're exposed to an antigen at the same time that they're able to bind. Plasmacytoid dendritic cells, as I mentioned, secrete interferon alphas. They make a lot of other things too. And they secrete exosomes with antigens, whatever antigens they've been taking up, that are cross-presented by other dendritic cells. Now that early phase of the innate immune response, that's in the first hours, that then leads to the subsequent adaptive immune response over the following days and weeks, where B cells now are secreting large amounts of antibody, and the PDCs and other dendritic cell types can induce antigen-specific T cells, including both the CD4 positive T cells that help this response and the CD8 positive T cells that are potentially able to kill infected cells or tumor cells. Now that's just at a high level. It turns out that depending on the structure of the TLR9 agonist, on the structure of the CpG DNA, you can push this one way or the other. And one class of TLR9 agonist called CpG-B agonists, the B stands for B cells, because these preferentially activate the B cell response. And they're very good at stimulating large amounts of antibody. They have less effect on inducing plasmacytoid dendritic cells. And the type of plasmacytoid dendritic cell activation that these induce is called P3 PDCs. These preferentially promote the CD4 T cells. They don't make much at all interferon alpha or drive the CD8 T cells as much. Now, this type of oligo is characterized by a linear structure like this and a phosphorothioate backbone. CpG 1018 is the CpG that's in an approved hepatitis B vaccine, Engerix-B. The idea is you simply swap this out for the normal adjuvant in the Engerix-B vaccine and enhance the response. So Engerix-B, the approved vaccine that many of us have received, consists of the hepatitis B surface antigen, which is the protective antigen that you want to induce antibodies to be protected. And as an adjuvant, as something that will cause the immune system to make antibodies to that, it uses alum, which is an aluminum salt that was developed more than 100 years ago. And it was just found by trial and error that it could stimulate antibody responses. So in Engerix-B, there's a 3-dose regimen. You get the first dose at day 0, a month later, and then 6 months later. And after those 3 doses, more than 80% of people achieve these protective antibody levels. Now, if you just switch alum for this CpG DNA, and all you need is 3 milligrams, that's in the approved vaccine. Keep in mind, how many milligrams do we use for antisense oligos and other applications? This is an exquisitely sensitive receptor mechanism to detect that CpG DNA. And the effect is profound, because after 2 doses of the Heplisav-B with the CpG added, already you've exceeded the protection level that you get after 6 months with the alum adjuvant. So you get much faster and more profound seroprotection. Early on, when these trials were started, there were concerns maybe this would induce autoimmune disease. But after this was approved in 2017, post-marketing surveillance studies in tens of thousands of humans have confirmed there really is no detectable difference in the safety of Heplisav-B compared to Engerix-B. So I think there's huge potential for using this type of CpG oligo to boost vaccine responses where you predominantly need antibodies. Now, what about using this for cancer? Well, you can boost antibody responses using this type of adjuvant for cancer. But the problem is that in patients with melanoma, high P3 PDCs have been associated with worse survival. And we took, at Coley, we took a CpG-B into phase 3 clinical trials in lung cancer, and it failed. And in general, the CpG-B and related CpG-C oligos that also have that phosphorothioate backbone, they have very little monotherapy activity in cancer. So I think think there are limitations to the application of this type of TLR9 agonist for cancer immunotherapy. Now, the other type of TLR9 agonist that I think is really very intriguing are the CpG-A. A stands for interferon alpha, because the defining characteristic of these TLR9 agonists is a profound activation of PDCs into a subset called P1 PDC. There's no other innate immune activator that's been published to induce a higher fraction of P1 PDCs or more interferon alpha than you get with CpG-A oligos like this. And that, of course, drives a CD8 positive T cell response. In patients with melanoma, high levels of P1 PDCs have been associated with improved survival, and I'm going to tell you about some of the early trials of the CpG-A. Now, what makes the CpG-A structurally different from the other TLR9 agonists? First of all, it has a native DNA backbone, and it does turn out during PDC differentiation, there's an obligatory cleavage step of the DNA template in order to get the maximal response. This G quadruplex structure seems to have a very important role too. These form G tetrads that stack into G quadruplexes. These will spontaneously self-assemble into multimers, and when you activate many immune receptors with a multimer compared to a simple linear ligand, you get a very different biologic response. And I'll just note, G quadruplexes have been reported to be present in the genomes of all human retroviruses and viral agents. So it would make sense that the immune system has evolved a way to distinguish that type of structure and make a stronger response to it. Now, because you have a native DNA backbone, you do have to worry a little bit about degradation. These can be packaged inside a virus-like particle, and that drug is called vidutolimod, containing this G10 there. And we were developing that at Checkmate Pharmaceuticals. And I just want to show you the results from the first patient in our trial at the 3 milligram dose level, where we were using intratumoral vidutolimod in combination with systemic pembrolizumab. This is a PD-1 blocker. And this trial was performed in patients where PD-1 blockade alone had failed to induce regression of their tumors. They had, or they relapsed afterwards. These were refractory tumors. So this patient had previously been treated with these various treatments and then presented with dozens of metastases. There were no other therapeutic alternatives at this point. And this was the primary injection site in the lower leg, 3 milligrams of this virus-like particle that I mentioned. That contains less than 1 milligram of CpG-A. And the dosing was weekly for 7 doses and then every 3 weeks. And within 5 weeks, looking at the tumor biopsies from baseline to the 5-week biopsy, this red indicates all of the CD8 positive T cells that are now flooding into the tumor. PD-L1 is a sign of inflammation, also indicating dramatic encouraging evidence there that we're getting the response we want. And then looking at the injected lesion, this is just the percentage of cells that are CD8 positive T cells, and also in a non-injected lesion. So we're getting evidence of a systemic effect here. And when you look at the lesions in the leg of this patient from baseline to 12 weeks and then out further, you can see that we're not just seeing regression in the injected lesion, and other lesions were injected during the course of therapy, but non-injected lesions are regressing as well. And that's really a key point, because when you're developing a cancer immunotherapy and it's intratumoral, you really don't have a drug if all you can do is cause regression of the injected lesions. You really have to achieve a systemic effect if you're going to benefit patients, and vidutolimod seems to do that. So this is what vidutolimod looks like. The CpG-A is aggregated into a nanoparticle that's then packaged just using the coat protein of a bacteriophage Q beta, and that protects it. This is not an infectious particle. And what happens when you inject this is this induces a very strong antibody response to the surface, as you might expect. This is very immunogenic. And those antibodies form immune complexes, and that's how these particles get into cells. There's no human receptor for these particles. The only way they get into cells is when you get immune complexes, and then they can be taken up through Fc receptors that take up immune complexes. And that's where the PDCs respond with this very strong interferon alpha secretion. Now, in mice, we did compare the VLP delivery to naked CpGA, and we saw better systemic responses using the VLP delivery. So there may be something about this uptake that improves the activity. But what really matters, of course, is the human clinical data. One of the studies was done in a setting called neoadjuvant melanoma. This is where you have very limited disease. It's very early, relatively small tumors. In that case, the pathologic response rate was more than half of the patients. And what's especially noteworthy is the relapse-free survival. In the patients who responded to treatment 2 years later, the great majority of them had not had a relapse. So there's now a controlled trial underway to try to reproduce those results. And then in more advanced melanoma, metastatic melanoma, where PD-1 blockade, which is the standard of care, has already failed to induce responses, we had two groups of patients. One received vidutolimod as a monotherapy without the PD-1 checkpoint inhibitor, and out of those 40 patients, we had over a 20% response rate. This level of response is unique among innate immune activators. There's no other agent that has shown that level of response. And this includes systemic responses. Some of the patients had liver, lung, and visceral soft tissue mets. And interestingly enough, 2 of these responders had previously been treated with an oncolytic virus, telimagene, in combination with pembrolizumab, had progressive response, then got vidutolimod as a monotherapy and had a response. So this is consistent with the idea that this is actually functionally different from an oncolytic virus. Now, the duration of these responses was less than 6 months. And what we think is happening is that the T cells that are induced by vidutolimod have the PD-1 checkpoint on their surface. And so unless you block that PD-1 checkpoint with pembrolizumab or another agent, you have a limited duration of response. In the arm of the trial with 98 patients who got pembrolizumab in combination with vidutolimod, we had a similar response rate, but the duration of response was over 2 years. So we're really getting a much longer response. This is the way that we would go forward in development. And we importantly saw similar tumor reductions in the injected and non-injected target lesions. And this included visceral metastases in some patients. We saw a shrinkage in bone, CNS, liver, lung, and spleen metastasis. And we've also identified biomarkers that help to predict which patients are going to respond to treatment and which patients are not going to respond to treatment. But just to show this key point about the shrinkage in the non-injected lesions, this is just for the responders in the trial. But there were only 1 or 2 responders in the trial where we saw regression in injected lesions, and we did not also see comparable regression in non-injected lesions. These are the patients who only had injected target lesions, and this shows the shrinkage. This is 100% shrinkage. And these are the patients who had non-injected target lesions that are shown in blue. And we give them the calculations here. But basically, you can see overall, there's a tendency here that it's pretty similar regression of non-injected and injected lesions. And these patients out here who don't have any orange bars, in that case, the injection was not given into a target lesion. It was given into a non-target lesion, which doesn't count for the measurement purposes under RECIST. So let me just take a big step back now and kind of summarize where I think cancer immunotherapy is at. I think it's generally agreed. Cancer immunotherapy requires tumor-specific CD8 positive T cells. There are approaches developing NK cell therapy for it, but this is really the main focus. Now, if patients have have T cells present in their tumor, and they're being held back by these checkpoints like PD-1 or LAG-3, and you add a checkpoint inhibitor, then that sometimes allows those T cells to attack the tumor. But in most patients, that alone is not enough to cause a regression of the tumor. And so one approach is to make exogenous CD8 T cells, like either tumor-infiltrating lymphocytes or CAR T cell approaches, and give those to patients. That's very complex. It's very toxic. And so another approach is in situ immunization. What we did in the patient I showed you, where we're injecting an innate immune activator into one tumor in a patient who may have widely metastatic disease. People have called this make cold tumors hot. And the idea is you can use the tumor as the source of antigens and let the immune system figure out which antigens to drive the T cells to. Now, all of these approaches rely on type 1 interferons. But really, as I mentioned before, interferon alpha is what is going to induce the T cell response that you want. Interferon beta is immune modulatory. And so when it comes to inducing interferon alpha to get this approach to work, you really need to activate this plasmacytoid dendritic cell. This is the major cell making that. They have a key role in tolerance versus immunity. And as I mentioned before, there are multiple subsets of these, but this one subset is particularly important, the P1 subsets, to make the interferon alpha, P3 driving more of a CD4 response. And so this may be related to the concept of Th1 versus Th2, but the implications for cancer immunotherapy are that we should try to optimize this P1 subset. That's what we want more than this P3 subset. And the only way to get that that's been reported in the literature so far is with TLR7 or TLR9 agonists. Now, I've already talked about the TLR9 agonists, so now let me just talk a little bit about TLR7 activation for this purpose. TLR7, like TLR9, forms a homodimer. Here are the two loops of TLR7. And with an RNA agonist, you can see it kind of clamps it between the two arms. There's UUU that fits ideally in the center, and then there's a second site, or a first site that binds guanosine, and a second site that binds this U-rich region. And of course, in our RNA, the U is modified. And approved COVID mRNA vaccines use pseudouridine in order to avoid TLR7 activation, because the interferon alpha induction suppresses the protein production from the vaccine. And now cancer vaccines, on the other hand, if you really want to induce this interferon alpha, you're going to need to use U in the vaccine or some other way of activating the interferon alpha production, or you're just not going to get the maximal CTL. You can still get CTL, but it's not likely to be maximal. Now, the other way to activate TLR7 is with small molecule agonists. Those only bind TLR7 at the first site. They don't bind at the second site. And biologically, that's very different. Now, these still have activity. They still can induce effective immune responses. And there is an approved drug, Aldera, that's approved for topical local therapy of early skin cancers. It is not systemically effective. And other small molecules that people have tried to develop for systemic therapy of cancer have repeatedly failed in the clinic. We do know that small molecule TLR7 ligands induce much less interferon alpha compared to RNA ligands, especially in immune or protein complexes. And so I want to make a prediction, although there isn't yet literature on this. I think that optimized RNA ligands for TLR7 should be superior to these small molecules for interferon alpha induction in cancer immunotherapy. And if there's one thing I'd like to encourage young scientists to go into, it's trying to develop these and figure out how to deliver these into tumors, not just by intratumoral injection, but through systemic delivery. That delivery is one of the major unsolved problems. But let me turn back now to RNA therapeutics more generally and remind you from the earlier slide, these are some of the best characterized receptors that can be activated by RNA therapeutics. If you're doing protein replacement therapy with mRNA, then you really don't want to activate any of those. And you can do that using modifications and rigorously purifying your nucleic acid. If you're making an mRNA vaccine for infectious diseases or for cancer, then you need to activate one or more of these. Any of these receptors can enhance an antibody response, but the CTL response really does seem to require, or is likely to require, the TLR7 activation. And the other thing, though, that's really interesting about stimulating TLR7 with U-rich RNA compared to TLR9 is that TLR7 is more broadly expressed in human immune cells. TLR7 is expressed in some myeloid cells, for example, myeloid-derived suppressor cells and tumor-associated macrophages. And these are populations of cells that have been associated with suppression of responses to cancer immunotherapy. So if we can develop RNA ligands that will stimulate maybe the same B-cell and PDC responses that you get with TLR9, but you add the stimulation of these myeloid cells, will this overcome immune suppression by those cells in patients with cancer? And of course, what other nucleic acid receptors should be activated in addition? You could design this to have a 5' triphosphate, for example. So I think these are some of the wide open areas for scientific research that I'm really hoping people will be pursuing in the coming years, because I think there's a great deal of promise in that. So the conclusions, what I really want to leave you with in terms of how to optimize innate immunity for cancer immunotherapy, I think there's very strong evidence that we need to induce interferon alpha preferentially to interferon beta. And in particular, diving more deeply, that we want to activate the PDC to differentiate into that subset that's been called P1 that secretes the interferon alpha. And the only ways to do that we know right now are with the TLR9, especially CpG DNA, or TLR7, especially the U-rich RNAs. Now, mRNA vaccines for cancer, some companies are using pseudouridine modified mRNA vaccines. Some companies are using uridine. So I think time will tell. We just need to see the data from those trials. I think everything is going to work a little bit. Some will work better than others. Oncolytic viruses are a special case here. Oncolytic viruses all rely on activating innate immunity against the tumor as a part of their mechanism of action. But very few of the groups studying oncolytic viruses have actually tried to optimize them and think about what effect are these having on interferon alpha production, because usually they try to avoid it. Interferon alpha suppresses the replication of oncolytic viruses. And so I think for those applications in cancer immunotherapy, we need to start thinking about the virus as not just its potential for infecting tumor cells, but also its potential once you've given repeated doses and there are antibodies to the virus and infection is now neutralized. Those viral immune complexes that will form with the antibodies, those could actually be very good, very effective TLR agonists that could help to drive these immune responses. And so in designing clinical trials, I think it's worth thinking about that. One of the other points that I've discussed with companies developing new agents in cancer immunotherapy is how do you pursue your clinical development strategy? I think we could think of vidutolimod as sort of the baseline for an innate immune activator, where we already have something as an innate immune activator that can induce monotherapy responses in patients where checkpoint inhibitors have failed. New agents going into development hopefully will also show that activity, or hopefully surpass it. And these responses, when they occur, should be systemic responses. If you're developing an innate immune activator that doesn't have monotherapy activity and doesn't induce systemic responses, I don't think you have a drug that is going to have much of a future. And finally, mouse data are not predictive of human results. Probably sound like a broken record. And with that, thank you very much, and I'm happy to answer some questions.
Yes, thanks a lot, Art, for this outstanding lecture. I think it was great to hear. So I will start off with a probably very naive question, but I really don't know. So I think you were mentioning that you have to induce interferon alpha rather than interferon beta. So, but what about interferon gamma? Because I always thought that this was like a regulator of immune responses and inflammation. So has
Yes.
this also a role in this whole?
Absolutely. Interferon gamma is extremely important. It's a type 2 interferon, and interferon gamma is critical for the efficacy of the T cells that are responding. It's also produced in very large amounts by natural killer cells. I sort of left them out to keep things somewhat simple. The earliest marker of a therapeutic response seems to be interferon alpha, but interferon gamma comes along later. When you have therapeutic responses, you should see interferon gamma, absolutely.
That was very clear. Yeah, and I think, just to say, if you have a question for Art, so please use the Q&A function. So then we go to the question from the Q&A in a bit. And I think, so this is an OTS webinar, so I think many people are interested in all kinds of anti-scent approaches. So can you elaborate a little bit about how this, because these anti-scent approaches have people use different backbones, different modifications, different strategies. You have gapmers that consist of RNA and DNA. You have naked oligonucleotides. So can we make a little bit sense out of this, or is it just impossible to classify this?
No, I think it is. I think, the nucleic acid therapeutics that we use can activate a whole range of different receptors, and to different degrees and in different ways. And for example, the phosphorothioate backbone has a lot of somewhat, people used to call them non-specific immune effects, some of which are mediated through the TLR9. I think the modifications can certainly suppress this activity. For example, not just the 5'-methyl modification on the cytosine, but using other 2' modifications greatly reduce the TLR stimulation. I think what's really helped the most in development of nucleic acid therapeutics has been optimizing the potency of the compounds. We've seen with siRNAs now, we're down at less than 1% of the doses that used to be required. And so that gets down to the level where you were no longer seeing a lot of those effects. Now, if you're developing a nucleic acid therapeutic for cancer immunotherapy, then maybe you want some of these stimulatory effects. And then being aware of them and knowing how to assay them could help you in your drug development.
Okay, so to have a quick question on this, question from Hassan in the Q&A. So does TLR3 get activated with siRNA?
Yeah, it depends on whose data you believe, I guess. And there are differences between mice and humans, apparently, on the ability and on the length required of the siRNA to activate TLR3. I think with the siRNAs that are in development currently and have been approved, it appears that they are not stimulating TLR3, but some of the early siRNAs could.
Okay, another question from Hassan. So can you elaborate a little bit more about the role of IL-6 in the immune response? And for example, how might NAT, so I assume nucleic acid therapeutics, might affect its response?
Yeah. So IL-6 is a part of the inflammatory response. You get IL-6 with pretty much any kind of immune stimulation, but you get it to different levels. So for example, with CAR T therapy, we're not activating any nucleic acid sensing receptors. And CAR Ts are associated with very high levels of IL-6 production, and it correlates with cytokine storm. In the trials of the various CpG oligos, we can see increases in IL-6 in the serum of patients who are responding. It doesn't correlate with whether they respond or not, but it's nowhere close to that level that is associated with toxicity. So I think IL-6 is a good example of a kind of cytokine where you need a little bit. Too much can be bad. And it's simply a sign that there is some kind of ongoing inflammatory response. It doesn't tell you whether that inflammatory response is going to be therapeutic.
I think it's very clear answer. So a question from Hans. Why hasn't viral evolution selected against G quadruplexes?
You know, I asked a virologist that, and they told me that they thought this was because the viral genome has to be so small. The virus, as a part of its replicative strategy, it has to form these G quadruplexes in order to initiate replication, transcription, reverse transcription, and these structures seem to be required for that. So it may be tough for them to avoid.
Yeah. Question from Bruce. Can vidutolimib be linked to tissue tumor-specific antibody FAB to target the drug with systemic delivery?
Absolutely, in principle. So this virus-like particle, the Q beta protein, it has solvent-accessible lysines that you can couple antibodies to, small molecules, carbohydrates. This has been reported in the literature, and I think there's a great deal of interest in those kinds of strategies.
Question from Robert. Thank you for the stimulating and intriguing presentation. Well done. However, here comes the question. How do the different interferon alpha subtypes play into the picture? And there was a study in 2020 showing that interferon alpha 2 and interferon alpha 9 are the only subtypes capable of completely controlling tumor outgrowth.
Yeah.
So
No, this is a really interesting question. Why do we have 13 genes for interferon alpha? There's no other immune cytokine or factor that we have 13 genes that have been maintained through evolution. I have to believe there's some role there. And as you say, some of these have been shown to have different biologic activities. There's a half dozen or so papers I came across when I was looking at this myself. I think the simple answer is we don't know. And I hope people will study this more because there have to be a lot more biology there to be discovered.
Okay, question from Hassan again. So I think in your conclusion slide, you mentioned that mouse data is not predictive, or animal data is not predictive. So what would be your suggestion?
Yes. So my suggestion is to use human cells in vitro. And I suggest starting with screening assays using just human PBMCs from normal donors. Those will contain all of the different immune cell types. And that's just as a starting point to see, compared to positive controls like a CpGA, you can buy the CpGA 2216 from Invivogen, or you can make it yourself, and you can get other positive controls as well. And then just test your agent against those controls to see what your interferon alpha induction is. And then we had a collaboration with Carlos Chan, this has been published, where Carlos is a surgeon and he was treating patients with ovarian cancer and other types of tumors where they develop peritoneal ascites, collection of fluids in the abdomen that contain tumor cells and tumor-associated immune cells. And Carlos showed if he took those cells, he could then add vidutolimod or other innate immune activators, and he could measure different levels of immune cytokine production in those patient samples. And I think that may be a very useful way to compare different agents for their ability to overcome myeloimmunosuppression. We've also talked with people about using tumor organoid culture systems. The problem is a lot of the tumor organoids don't seem to contain the myeloid cells that would be of greatest interest. So that's kind of where we're at. I think we probably need better systems for this.
Yeah, so that's always my question, right? So is it fair to test your agent within a control cell or within healthy cells, whether in principle you want your agent to act on a tumor or in a disease cell, right? So how predictive are healthy cells for that?
Yeah, they're not perfectly predictive. We know there were some very nice studies done by Gunter Hartmann's group some years ago where they took tumor biopsies from patients with head and neck cancer, and they actually took the primary PDCs in those tumor biopsies and tested them in vitro for their ability to make interferon alpha in response to CpGA. And what they showed was the patients who had earlier stage head and neck tumors had in general better interferon interferon responses than the patients with more advanced tumors. That's a very difficult kind of model to use for drug development. But I think it makes a key point that we have to be aware of, that the more advanced a tumor is, the more mechanisms there are going to be that are suppressing these responses. And if you do all of your assays in normal cells, you're going to miss that.
Yeah, I think it's a very good suggestion as well. So a question from Elena. Thanks for the amazing lecture. What is the role of delivery systems and the route of administration in response to immune stimulatory nucleic acids?
Yeah, I think it's really critical. You know, I skipped over it, but there have been studies showing that you can take CpGB oligos and formulate them in nanoparticles and cause them to aggregate, and their biologic properties change dramatically from their linear arrangement. I think that delivery systems are going to be key. One of the limitations we faced in developing vidutolimod at Checkmate was that there's a limited number of patients that you can easily do intratumoral injections. And so if the field can come up with vectors or targeting ways to get these into tumors by systemic administration, IV, sub-Q, that's going to be a huge advance. I know a number of groups are working on that, and that could really, I think, transform cancer immunotherapy.
A few questions left, so I think we have 3 minutes, so I think we can answer them all. At least we're going to try. Question from Nagis. Can we use interferon alpha alone for cancer treatment in the form of purified protein or antibody fusion protein?
Yeah, you can, but it doesn't work very well. It's approved for treating a few types of tumor, but the efficacy is quite limited. And the reason why I think it's so much worse than activating the PDC is twofold. First of all, high levels of constitutive interferon alpha cause T-cell exhaustion. The PDC makes the interferon alpha in a pulsatile manner. You get a big pulse that blasts the T cells into proliferating like crazy, and then it wears off. And by doing that once a week, you can get this enormous acceleration that you don't get with recombinant factor. Secondly, the PDCs also take up antigens from their environment and secrete them in exosomes when they are stimulated through TLR9. And those exosomes are taken up by other dendritic cells, like conventional dendritic cells, that then cross-present the antigens to T cells. PDCs aren't very good antigen presenters. They really need to collaborate with other cells, and there are cell-cell interactions there that you just don't get with recombinant interferon alpha.
Question from Louisa. Which nucleases are relevant in processing RNA and produce the RNA fragments that ultimately stimulate the TLR7?
Yeah, that's a good question. You know, I don't think we know. I think DNase2 is clearly the nuclease for the CpG DNA. For RNA, we can infer there must be likewise nucleases that are important. I just don't know which ones they are.
Yeah. Question from Joey. Do we ever check platelet counts on responders versus non-responders? Platelets express TLR9 and can drive interferon alpha production from PDC.
Yeah, there was no significant difference in the platelet counts in responders and non-responders. And we did try to look for any direct effect on platelets. We couldn't detect it, but I don't know if we were, working with the right cells or not.
Okay, and then I think we're perfectly into time because we have one question left from Teresita.
Actually, I can read it too. It looks like it's a long question.
Since 1999, I've been preparing a suspension of oligonucleotides of biological material in people. There is a reversal of all autoimmune diseases showing a state of healing. Brain tumors decrease in size. People with prostate cancer have been cured with the help of this product. Patients with colon cancer remain without manifestation of the disease. The product is drinkable, and I do not see that it produces adverse reactions or any other adverse events. Can you comment on this?
I'd be interested in seeing the data. I think, clearly nucleic acids can have a whole range of immune effects. And we are still learning how those work. So it sounds interesting.
Okay. And with that, I would like to thank Art for this excellent presentation. I enjoyed it a lot.
Thank you.
I would like to thank Erin for co-hosting. The next OTS webinar will be in 2 weeks from now. And then we have Dr. Timothy Yu, and he will be presenting on the N-of-1 collaborative. Also, I think it is part also of the OTS or something that also the OTS initiated. So I think it will also be a very nice webinar to watch. So Art, thanks
Thank you
once again. I really